So the second thing I want to talk about that's a component of this, and I'm kind of a molecular biologist and a modeling guy, is this side of the diagram. I spend a lot of my time here as well. And that is taking a disease gene that you know and then trying to make a model of it, and then hopefully see if you can bring it towards intervening on that model. And so the one that I focus on is this Charcot-Marie Tooth disease, and you heard a little bit about it. This individual here is one of my adult patients. He presented as a child, and he has wasting of all the muscles here in the legs, and then in the hands. I think you see sort of the scalloping of those muscles. That's because those nerves have degenerated and died back. And this is another patient who, again, she has loss of the muscle in her leg. And the long story short is one of the more common causes of this type of disease are mutations in this protein called mitofusin-2, or MFN2. And this protein sits on mitochondria. So mitochondria are the powerhouse of the cell, in a sense. And these things actually have to be distributed all up and down the nerves in somebody's body. And so we went about trying to model this disease, like I'd said, in a dish. And what we do is we take out rat neurons and we put them in a dish, and then we generate viruses that actually allow these neurons to express your disease protein together with a marker, and that's red here for these mitochondria. And we actually look at them under a microscope, and really what we did was we just saw what happened. And this is in a normal setting, so this is a normal nerve. I think you can see that. So there's these little mitochondria here, right? And they're just zooming up and down this nerve. Okay, this is a nerve, an axon sitting in culture. This is the same one that would go from your back all the way down to your toe, okay? And so this is in the normal case, and the mitochondria are happily marching all along this axon. Now, if we put in the mutant form of this protein, I think that one's going to stop in a second. It's a long one. You see two things. One is that they're not the same in terms of their shape. They're actually really small, and they're not moving very well. So the only one, you get this kind of very wandering guy here, and he makes his way over. But that's really important. And what we think is happening, we've gone through a lot of work to show that this protein, this mitofusin-2 protein, sitting on the outside of the side of the mitochondria is really important for attaching to the microtubules that allow these mitochondria to move up and down these axons. And the idea is that the reason why, and I think it's on the next slide, you have problems here, and this with the movement of these things, towards the end of the line here, you don't have enough of them. And or they're just not working right. And so you have problems in regulation of calcium homeostasis and other things, but it really just leads to degeneration of these axons, and then the muscles shrink, and the patient is weak. So this is just, I brought this up as an example of how we sort of go about this right now. And we use a lot of rats and viruses, and every time I present this data, someone says, well, how do you know whether that's happening in the patients? And I don't. We've been trying to figure that out. It's extremely hard to look at a live neuron in a real patient, and also early on when you might want to see it. And so what we've been doing, and as really an extension of the Neuromuscular Genetics Project, is developing another project, which is the fibroblast and induced stem cell project. So this is a new technology. It was not invented here. It's a really important technology, and I think it's going to change the way we model human diseases. And that is, we can take cells from a patient. In this case, this was a patient who has an MFN2 mutation, and these are her fibroblasts. And then we treat those with a series of viruses to turn them into what are called induced pluripotent stem cells. So they're stem cells, meaning they can turn into any other kind of cell, but they're not embryonic stem cells. They're not derived from an embryo. They're derived from a patient's fibroblasts. So they're genetically like her. What we can then do, as I said, I want to study the neurons, but I can't really take hers out and put them in a dish. But what I can do is turn these into neurons. And then I can actually ask the same types of questions that we had been doing before. We can put in other markers. We can probe these, and do we see? Does it look normal? Does it look abnormal? And if it does, can we fix it in a dish? And so this is really, this is me giving one of my 3 biopsies. We do the non-sun exposed skin of the thigh, or buttock, if you like. I did 3 before we got it to work, but it works great now. So anyhow, so we're doing this, and we're collecting these because we really think that this is going to be important. So we can take the skin fibroblasts, even if we're not going right now, and we can derive them from the patient, so we can freeze them down, and we can then study them ourselves. We can tell people we've got them. Why don't you come get them, and we'll give them to you so you can study your interested type of muscular dystrophy that you would like. So we've already got 211 lines. Patients, it's really not a very invasive procedure. There might be better ways of getting these in the future. Some people are talking about trying to get cells to turn into stem cells from blood cells. It's not working as well right now, so we're sort of going with the gold standard. It's a 3-millimeter punch biopsy. It's a very standard kind of thing that you would do with the dermatologist. So that's really where we're going with this. We've been able to turn them into induced pluripotent stem cells. We've started to turn them into neurons. And we really want to get this out there that we're doing this, and we want other folks to start working on it with us. Because you may have heard recently about the heart, and it turns out it's a muscle, right? And a lot of our patients have heart problems. And maybe you guys would like to model some of the diseases that are inherited in your patients in a similar way. I think that we're trying to build the infrastructure to do that, and the CDI has really been instrumental for letting a young punk like me get that kind of thing started. So summary is, these diseases, as I said, are untreatable, and they are an important cause of disability and premature death in both children and adults. And this project really provides an infrastructure for studying these here at WashU for both gene discovery and therapeutics development. And like I said, the things where we're going, I think we're going to do very well in terms of identifying essentially very high numbers, if not all the gene defects in our patients very soon. And likewise, we want to correlate that with better modeling systems to understand their diseases and doing that with induced pluripotent stem cells. And I've already mentioned it many times, but I really want to thank the CDI for basically gambling on a guy like me and saying, I think you're going to do something with this. So that's really the end. Thanks.